Let's now proceed to talk about protein function. Proteins can be categorized based on the job that they need to perform in a living organism. One of the functions that proteins can have is actually the catalysis of biochemical reactions. Proteins that have this job are known as enzymes. Proteins can also have a structural function. And when you have proteins that have a structural function, is because there actually are structural material. Some examples of this are going to be collagen and keratin. Proteins can also have storage function. There are a number of proteins that provide a way to store small molecules or ions. Proteins can also have a protective function. For example, antibodies are a type of protein that are important in the protection of the body from disease. The next function that a protein can have may be regulatory. There's going to be a series of proteins that are called hormones, which specifically are going to be responsible for different processes that regulate the body. Proteins can also be involved in nerve impulse transmission, and these proteins behave as receptors of small molecules that pass between the gaps, in other words, synapses that separate nerve cells. Proteins can have movement function. These proteins are found mostly in muscle, and they are going to be present in long filaments, and they are going to allow muscle to basically contract, which is one of the functions. Lastly, we can have proteins that actually have transport function. Transport function for proteins basically are the proteins that are able to specifically transport small molecules or atoms, specifically in the body, by binding to the small molecules and ions in order for them to move through the body. Specifically, I mentioned first that there are some proteins that have catalytic ability, and I mentioned that they are called enzymes. Enzymes, because they are going to be present in the majority of biochemical reactions, we can actually talk about them in terms of a chemical reaction. So, in chemical reactions that are driven by enzymes, what we're going to have is the enzyme acting as a catalyst. And you may remember from previous chapters that catalysts are going to be substances that are responsible to increase the rate of a chemical reaction by lowering its activation energy. In biochemistry, we don't call reactants by that word. We tend to talk about them in terms of substrate. But understand, substrate and reactant is the same thing. It's basically the substance that is going to undergo a chemical change. When it comes to enzymes, we can have enzymes that have absolute specificity. In absolute specificity, it is a characteristic of an enzyme that it acts on one and only one substance. So whenever you have an enzyme that only has one specific substrate, then we say, oh, this enzyme is going to have absolute specificity. Some enzymes have relative specificity. And enzymes that have relative specificity basically implies that these enzymes are going to act on several structural related substances. Since enzymes are going to catalyze reaction, we can actually categorize them based on the reaction that they're catalyzed. As part of this course, we need to know the categories that we have for the classification of enzymes, starting with oxidoreductases. An enzyme that is considered an oxidoreductase is going to be an enzyme that catalyzes reactions that are involved in oxidation reduction. When we have an enzyme that is called a transferase, it's going to transfer functional groups between a substrate or different substrate to form a product. Enzymes that are called hydrolases are going to be enzymes that perform hydrolysis reactions, meaning that is going to take that substrate and cut it with water. Enzymes can be classified as lyases because upon the chemical change of the substrate, they are going to add double bonds or they are going to do the reverse of that, meaning that it may add atoms, but your substrate must start with a double bond. Next, we have enzymes that are known as isomerases. And isomerases are going to be enzymes that create constitutional isomers overall. So if when you compare the substrate to the product that was catalyzed by the enzyme, the substrate versus the product are constitutional isomers of each other. And lastly, we have ligases. Ligases are going to be enzymes that form bonds, but specifically require energy to do so. That's why we have this molecule that is called ATP. So just think of ATP as an energy molecule. And in order for a ligase to do its job, it needs to basically break bonds in ATP to input energy in order to do bond formation. So energy is required by ligases in order to do their job. Since enzymes are going to be the biomolecules that catalyze biochemical reactions, we can actually write them as an overall reaction. And this is the general reaction for an enzyme catalyzed reaction. We have an enzyme interacting with its substrate, in other words, the reactant. And when they come together, they make the enzyme substrate complex. Notice that this specifically is reversible. But because in a way, in an enzyme catalyzed reaction, the enzyme and the substrate have to actually follow the collision theory for reactions, meaning the enzyme and the substrate have to meet. They need to do it with enough activation energy. They need to come together with the correct conformation in order to make the ES complex. Once the enzyme substrate complex, remember the E is for enzyme, the S is for substrate. That's why we call it ES complex. Once this has formed correctly, then the enzyme is committed at forming product. But because enzymes are catalytic, they are regenerated. They do not get destroyed. They do not get change. Overall, they are regenerated at the end of the reaction so they can go back and act on a brand new substrate when necessary. This general reaction for enzymes happens to be located in a specific part of the enzyme, and that is called the active site. So all the chemistry that happens in an enzyme, it occurs at the active site of the enzyme. Enzymes have different mechanisms of actions, and there's actually two theories that explains the different mechanisms of actions for specifically how enzymes act. The first one is the lock and key model. The lock and key model, or the lock and key theory, is a theory that proposes that enzymes' specificity for its substrate has a specific shape in which the substrate has to fit the shape of the enzyme active site, like the lock and key. So basically, the active site has exactly the shape of the substrate, and that's how they interact. When we look at image A in figure 12.24, we can see a drawing of that, how that works. The other theory that exists for enzyme mechanisms is the induce fit theory or induce fit model. In this theory, what happens is that enzyme action is proposed to be more flexible. In this type of mechanism action for an enzyme, what happens is that the active site of the enzyme actually accommodates a incoming substrate, but in turn, the active site is not exactly the shape of the substrate. What happens is that when the substrate comes and fits into the active site, the enzyme itself has like a conformational shape, and it changes its shape to actually wrap itself around that substrate. So, out of the two theories, induce fit theory or induce fit model is more dynamic. It's not as rigid as what we have for the lock and key theory, which the shape of the active site, it is exactly the shape of the substrate. In the induced fit model is just an active site that can accommodate the substrate and have conformational changes to actually adjust to the shape of the substrate. When it comes to enzymes, sometimes enzymes need help. And when we have enzymes that need help, essentially what happens is that enzymes, some of them, may require coenzymes or cofactors in order for them to be able to be catalyst. Overall, an apoenzyme is going to be an enzyme that is inactive. Apoenzymes may interact with either a coenzyme or a cofactor in order for them to make a holoenzyme. And holoenzyme is the active form of an enzyme, and that's what can interact with the substrate. Coenzymes are going to be organic molecules required by an enzyme in order for them to be active, while cofactors are going to be a non-protein molecule. So essentially, they're going to be ions that may include magnesium ion, manganese ions, iron, copper, zinc, calcium, or cobalt ions that are going to be present with the apoenzyme in order to make a holoenzyme. Understand that coenzymes can be specifically coming from vitamins. When it comes to examples of vitamins that act as coenzymes, all of these are summarized in table 12.6. As we have seen so far, enzymes are very important proteins in living organisms because they are true catalysts. And as I mentioned, the way that they increase the rate of the reaction is by overall lowering the activation energy in a reaction. Enzyme activity refers to the rate at which an enzyme catalyzes a reaction. Enzymes can actually have a turnover number, and the turnover number refers to the number of molecules of substrate that is acted on by one molecule of enzyme per minute. So you have an idea of the turnover number per minute of some enzymes, you can look at examples in table 12.5. Enzyme activity can be affected by different factors. Some of them include the concentration of enzyme, the substrate concentration, the temperature at which the enzyme reaction occurs, or even the pH of the solution where we have our enzyme. Let's first look at how enzyme concentration actually affects enzyme activity. Since enzyme activity is rate of an enzyme catalyzed reaction, when it comes to enzyme concentration, let's take into account that the following relationship is true if we have constant concentration of substrate. So when we have constant substrate concentration, what the relationship is between increasing enzyme concentration versus the rate is a linear relationship, meaning as we increase the amount of enzyme, overall, our enzyme catalyzed reaction gets faster and faster and faster. But if you think about it, it's kind of like having many workers doing a job. So the more people that come and perform the job, then that's going to make the job go faster. That's basically an analogy that I utilize in order to rationalize why that is. But what if we keep enzyme concentration constant and we instead change substrate concentration? And let me point out that the reason why we have Miss Maud Menton in the previous slide is because she was the one that did a lot of contributions into our current understanding of enzyme catalyzed reaction. You can see her picture in figure 12.26. So all of this work in understanding enzyme catalyzed reaction was done by a woman in science. Going back to what about if instead of changing enzyme concentration, we actually change substrate concentration? So if we have a constant enzyme concentration, then what happens overall to the rate? Well, it is interesting to note that initially the rate is responsive to increases in substrate concentration. However, at a certain concentration, you can see like around here, roughly, the rate level remains constant. And that's why when we have an at constant level of enzyme concentration, if we keep on increasing the substrate, what happens is that the rate of the reaction maxes out. This is symbolized by what we call in biochemistry Vmax, which is basically the maximum rate of your enzyme catalyzed reaction. This happens because at some point the enzyme is saturated with substrate and it cannot work any faster during the conditions imposed. So if we go back into the analogy that we had first, so understand that in this case, it's like you have a certain amount of workers for a job, and then you have incoming jobs, but you only have a constant number of people. So it doesn't matter that you keep on increasing the jobs that these people can do. At some point, that you're going to saturate them with work, and they have reached the speed that they can actually complete the job in. And we can see that this behavior also happens in enzymes. When it comes to temperature, specifically, enzymes have what is called an optimum temperature. An optimum temperature is going to be the temperature at which your enzyme has its highest activity. For the enzymes in the human body, that's going to be specifically 37 degrees Celsius. In other words, body temperature. It is important to know that for enzymes, if you lower the temperature or you increase the temperature, both affect enzyme activity. So that's why you see that the curvature of our graph starts decreasing away from the optimum temperature. It doesn't matter if we decrease it or if we increase it. Now, if you decrease the temperature from the optimum temperature of an enzyme, what happens is that in colder temperatures, then you're affecting the kinetic energy of the enzyme. So basically means that if you cool it down, it's not going to be able to move as much. So that's why we see a lower activity. On the other direction, meaning if we increase the temperature from the optimum temperature of the enzyme, what happens is that this causes denaturation. And denaturation is basically the event at which proteins, or in this case, enzymes, are going to start unfolding. And because they are unfolding and losing the shapes, then that is going to affect their activity. In denaturation, essentially what you may be affecting within an enzyme may be its quaternary, essentially tertiary or secondary structure. Lastly, pH can affect enzyme activity. And when it comes to the effects of pH on enzyme activity, it is similar to temperature in which pH has an optimum pH value. Now, different from temperature, the optimum pH for enzymes, it is not necessarily physiological pH. Enzymes can have different optimum pHs. You can see in the table in the bottom of the slide some examples of enzymes at their optimum pHs. You can observe that optimum pHs for enzymes can be in the acidic range, in the neutral range, but even in the basic range. By definition, the optimum pH value simply refers to the pH at which your enzyme has the most activity.